Hello, and welcome to this Microbiology Bio 203 video lecture for Chapter 15 of your OpenSTAX book, Microbial Mechanisms of Pathogenicity. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for Chapter 15 include being able to describe the characteristics of infectious disease and the mechanisms by which microbes cause disease. We begin today with some definitions. A disease is an abnormal state in which the body is not functioning normally. Diseases are caused by pathogens, or simply put, anything that can make you sick. Pathogenesis is the development of disease. Infection is the colonization of the body by pathogens. Signs and symptoms. A sign is a change in body that can be measured or observed as a result of a disease. An example might be body temperature or blood pressure. Signs are usually objective measurements. Symptoms are a change in body function that's felt by a patient as a result of disease, such as feeling down or tired. These are often subjective measurements. A syndrome is a specific group of signs and symptoms that accompany a disease. Classification of diseases. Diseases can be classified as communicable, contagious, or non-communicable. Communicable and contagious diseases can be spread from one host to another. The difference between them is the ease with which that spread is accomplished. Non-communicable diseases are diseases that cannot be transmitted from one host to another. Iatrogenic disease is a disease resulting from a medical procedure. Nosocomial diseases are diseases acquired in hospital settings. Acute diseases are those where symptoms develop rapidly, while chronic diseases have symptoms that develop slowly. A latent disease is a disease with a period of no symptoms when the pathogen may be inactive or dormant. Periods of disease. When infected by a pathogen, there is a natural life cycle of the pathogen that will occur in the host that begins with an incubation period, followed by a prodromal period. Then, as products or the sheer presence of the pathogen increases, illness ensues. Illness is the most severe sign and symptom period of the disease. Through the activity of the immune system or treatments from outside, the microorganism or pathogen making you sick will begin to decline in numbers. This will take us to the decline and convalescent stage of disease. Koch's postulates. You may remember a man by the name of Koch from a previous video. Koch's postulates are used to connect a specific disease-causing organism to a disease. There are, however, a few things that must be taken into consideration for Koch's postulates to be considered valid. First, the suspected pathogen must be found in every case of disease and not be found in healthy individuals. Second, the pathogen must be isolated from the diseased host and grown in pure culture. Third, a healthy test subject infected with the suspected pathogen must develop the same signs and symptoms of disease as seen in postulate number 1. And finally, the pathogen must be isolated from the inoculated animal and must be shown to be the original organism. This is an illustration of the application of Koch's postulates. While Koch's postulates are effective in attaching or connecting a specific pathogen to a disease, there are some times or potential opportunities where it is less than effective. Some pathogens may be part of the normal microbiota, like H. pylori. Individual responses to the same pathogen might be different, making it difficult to connect or legitimize the connection between a pathogen and a disease. Some pathogens can cause several disease conditions, like Streptococcus pyogenes. Some pathogens can't be cultured, and some pathogens cause diseases only in humans. Molecular Koch's postulates. As an extension of Koch's postulates, a molecular version was developed by Falkow. Here, the phenotype, or sign or symptom of the disease, should be associated only with pathogenic strains of a species. Inactivation of the suspected genes associated with pathogenicity should result in a measurable loss of pathogenicity. Reversion of the inactive gene should restore the disease phenotype. Pathogenicity and virulence. Pathogenicity is the ability to cause disease, while virulence is the extent of pathogenicity. Virulence can be expressed or compared using an ID50 measurement. ID50 stands for infectious dose. The infectious dose is the number of microbes that are able or needed to infect 50% of the population. The lower the number, the more infectious. A graph like this is used to determine LD50, which is lethal dose 50, by plotting a pathogen concentration against the percent of infected test animals that have died. LD50 is an extension of ID50. In LD50, this is the minimum dose required to kill 50% of the population that it's tested against. Here are some examples of individual pathogens and their ID50 measurements. Remember, the lower the number, the more dangerous the organism. Types of pathogens. Primary pathogens cause disease independently of the local microbiota or immune response, while opportunistic pathogens can cause disease when the host is susceptible. Opportunistic pathogens may be normal microbiota in certain circumstances, but when given the opportunity to change their ecological niche, they can become pathogenic. Organisms that live in harmony on the skin can be dangerous inside the body. Diseases by extent of host involvement. Local infection. Local infections involve pathogens that are limited to a small area of the body. Systemic infection is an infection throughout the body. A focal infection is a systemic infection that began as a local infection. A primary infection is an acute infection that causes the initial illness, while a secondary infection is an opportunistic infection after a primary or predisposing infection. Stages of pathogenicity include exposure, adhesion, invasion, and infection. Portals of entry. Pathogens can enter the body through mucous membranes, the respiratory tract, the GI tract, the GU tract, and conjunctiva. They can enter through the skin, usually through open wounds, or directly into tissues. Torch pathogens can pass the placental barrier and infect the fetus. Listed in the table are the torch pathogens and the diseases that they cause. Take a moment to pause the video and familiarize yourself with each before moving on. Adhesion. Adhesion is the capability of pathogenic microbes to attach to the cells of the body using adhesion factors. Adhesins bind to receptors on your host cells and include things like the glycocalyx and fimbriae. Invasion. Invasion is the dissemination of a pathogen throughout local tissues or the body. During the invasion, toxins or enzymes are used to penetrate the cells. Virulence factors to protect them from the immune system are also employed. Infection. Local infections, again, are limited to a small area of the body. Systemic, throughout. Focal, a systemic infection that began as a local infection. Primary infection is an acute infection that causes the initial illness, and the secondary infection is the opportunistic infection after a primary predisposing infection. Portals of exit. Portals of exit are similar to, if not identical to, the portals of entry. Virulence factors. Virulence factors involve the extent and severity of the disease and depend on the encoded genes in the pathogen that is proliferating within the host. Some examples of virulent factors and adhesions are listed for you here in this table. Pause the video and take a moment to familiarize yourself with the pathogen, the disease, the adhesion, and attachment site. Invasion through the bloodstream. Bacteremia is the term we use to describe bacteria in the blood. Toxemia is the term used to describe toxins in the blood. And viremia is the term used to describe viruses in the blood. Septicemia describes growth of bacteria in the blood, while sepsis is the toxic inflammatory condition due to the spread of microbes, mainly bacteria and their toxins. Sepsis can lead to septic shock. Vascular permeability will increase, leakage from the blood vessels, your blood pressure goes down, and circulation is impaired. This is a table of exoenzymes, listed by class, with example and function. Take a moment to familiarize yourself with the exoenzymes listed here before moving on in this video. Toxins. Toxins are substances that contribute to pathogenicity. Toxogenicity is the ability to produce a toxin. Toxemia is the presence of toxin in the host's blood. A toxoid is an inactivated toxin used in a vaccine. Antitoxin is antibodies against a specific toxin. Toxins can be exo or endotoxins. This table will provide you with some examples of exo and endotoxins. Take a moment to review this table before moving on. This is an example of a lipopolysaccharide endotoxin. You will not be tested on its molecular structure. This table will provide you with some examples of exotoxins. Take a moment to pause this video and review the type, the pathogen, the exotoxin, and its effect before moving on. This is an example of exotoxins or AB toxins as they're produced in a cell. Exotoxins may be membrane disrupting or superantigen. Membrane disrupting toxins will lyse the host cells, while superantigens will cause intense immune responses due to the release of cytokines from the host cells. Evasion of the immune system. It makes perfect sense that in order for a pathogen to complete its life cycle, it will need to, in some way or another, evade our immune defenses. Capsules can protect pathogenic organisms from phagocytosis, while proteases can protect against components of the immune system. M proteins resist phagocytosis, opoprotein inhibits T helper cells, and mycolic acid resists digestion. This is an illustration of a capsule and proteases. Other mechanisms of resistance against the immune response include coagulase, kinase, and antigenic variation. Viral virulence or adhesions. This table will provide you with a summary of pathogens, diseases, adhesions, and attachment sites for a variety of viral agents. Take a moment to pause the video and review this table before moving on. Antigenic variation in viruses. Antigenic variation is a tool that some pathogenic viruses, if you'll allow the human characteristic, employ to protect themselves against a host. Some enveloped viruses, like the flu, have antigenic variation. Point mutations are often the cause of antigenic drift. Gene rearrangements cause antigenic shifts. This not only helps the virus avoid the host defenses, but can also help the virus to infect new and different cells. Virulence factors in fungi. The virulence factors in fungi are similar to those previously described. They include adhesions, exozymes, capsules, toxins, and antigenic variation. Virulence factors in protozoa, again, are similar to those previously described and include adhesions, toxins, antigenic variation, survival in phagosomes and capsules. Virulence factors in helminths. Helminths are multicellular parasitic organisms, worms, and their virulence factors depend on conditions needed in order to help them penetrate host tissues. They can produce proteases to degrade skin proteins, evasion of the immune system, and depression of the immune system. And this concludes our coverage of Chapter 15, Microbial Mechanisms of Pathogenicity.